Greetings, viewers. I want to welcome you again to my YouTube channel. In today's class, we're going to be talking about one of the parasites in platyhelminthes. Don't forget that we introduced platyhelminthes as flat worms, and we'll talk about their characteristics. Now, one of the foremost parasites in platyhelminthes is a parasite we call the Schistosoma. Schistosoma is a very popular parasite. And let's start by talking about the scientists that discovered this parasite. This parasite was discovered by a man we call Theodore Bilhaz in Cairo, in Egypt. That was where this parasite was discovered in the mesenteric vein of a young man on autopsy. Now, having known the person who discovered this parasite, it is also important we also explain, or that I also explain, the fact that the former name for this parasite was not even Schistosoma. The former name was Bilharzia. Bilharzia. Now, it was called Bilharzia because of the name of the scientist who discovered it, and his name, like I said, is Theodore Bilhaz. So, from this word Bilhaz, the parasite was now given a name, now called Bilharzia. Now, the name of the disease it causes is called bilharziasis. Bilharziasis. So once you add ziasis, yeah, bilharziasis, that becomes the name of the disease. However, why is it now called schistosoma? Now, further discovery or further research on this parasite, was done by some other scientists, and they now discovered that the male of this parasite, has a canal that is called gynecophoric canal, and with this canal, the is a kind of receptacle through which they carry the females for easy mating because of the limitation imposed on them as a result of their parasitic mode of life. So the word schisto means split, split. The soma means body. Schisto means split, soma means body. So, what am I saying? It means that the body of the male schistosoma is split into two with this canal we call gynecophoric canal. So, if we have this as a male schistosoma, then they have a canal here that splits it into two, and this is where they carry the female. Now, with this introduction, we can now go into further details about this parasite. And these further details may mean that I talk about the different species we have in this parasite. We'll have different species, but the five major species are the ones I want to talk about. We have the one we call S as Schistosoma. I'm not abbreviating it. S. Japonicum. We have S. Mansoni. We have S. Mekongi. We have S. Intercalatum. We now have S. Hematobium. So these are the five main species of this parasite Schistosoma. We'll go into them again. The first one is Japonicum, we have Mansoni, we have Mekongi, we have Intercalatum, we have Hematobium. Now, there is one major difference between these parasites. Why this one is usually found within the veins of the bladder, that is Hematobium, then the Japonicum, the Mansoni, Mekongi, and Intercalatum are usually found within the veins of the intestine. That means that if you want to diagnose this parasite, of course, the eggs of these four species will find the feces of the host of the or probably the individual who is suffering from the disease, whereas the eggs of hematobium will be found in the urine of the individual who is suffering from the disease. Okay. Having given this introduction, let's now go into the life cycle of this parasite. The life cycle of Schistosoma. Now, we have that this parasite is a heterogeneous parasite. And what does it mean to be a heterogeneous parasite? It means that the parasite requires two hosts to complete its life cycle. And the life cycle is also being completed in an aquatic environment. It's also an important point you need to note. Now, the parasite begins by the penetration of the infective stage, which is called a saccharia. Now, I want us to understand this. There's what we call passive transmission and active transmission, and I think I've explained this in the previous classes. When we talk about active transmission, which is a type of transmission that occurs in this schistosoma, that means that the parasite on its own penetrates through the unbroken skin of the host. And this usually happens while the individual is swimming in water. As the individual is swimming in water, then the saccharia can now penetrate into the host. The saccharia has a shape that looks like this. It has a shape that looks like this, and it's also said to be bifid. It's also said to have a bifid tail. That means we'll have two different extensions in the tail. This is how the saccharia of Schistosoma looks. Now, like I said, the life cycle begins by the penetration of the saccharia. Now, as the saccharia penetrates into the host, then it joins circulation. First of all, it's being taken into the heart. Then from the heart, it joins what we now call the pulmonary circulation into the lungs, and from the lungs back to the heart again. From the heart again, it is now pushed through the portal vein into the liver. And it is the liver that they mature into adult. Please don't forget this. They mature into adults in the liver. And once they mature into adult, that is when they begin to separate. Members of those four species I mentioned will now migrate into the vein or the intestine, whereas members of the species which is known as hematobium will now penetrate into the vein or the bladder. Now, that is where they lay their eggs, and as they lay their eggs, of course, when the individual, which is now suffering from schistosomiasis, that's the name of the disease, when the person now passes out feces, now the eggs or the first four species I mentioned will now pass out to the feces into the external environment. And when the person or another individual, the same person, urinates, then the egg of hematobium will now pass out through the urine into the external environment. Don't forget that I said that the life cycle of this parasite is perpetuated within the aquatic environment. That means that if that environment is not aquatic, then it cannot complete its life cycle. So it means that it requires somebody who defecates in the river or in the water body, or somebody who urinates in the water body. And as the individual urinates or defecates in the water body, what now happens is that the feces or the eggs are being released into the external environment. As the feces are released into the external environment, then the eggs hatch in this aquatic environment. As they hatch, the eggs will now metamorphose into what is now called the meracidia. Now, this meracidia penetrates the host. The eggs metamorphoses into what is called the meracidia. Meracidia. Then this meracidia penetrates the snail intermediate host. Now, in the body of the snail intermediate host, the meracidia, transforms into what we call the sporosis. Usually, we'll have two different stages in this process. We'll have sporosis 1 and we'll have sporosis 2. We'll have the sporosis that looks like an egg, not yet formed saccharia. Then from this, we now have the second stage of the saccharia that is now looking like, sorry, the second stage of the sporosis that is now looking like a saccharia. Now, from this sporosis, it now metamorphoses into the last stage, which is saccharia, which is in the fertile stage. And once it forms saccharia in the body of the snail, then the saccharia also leave the snail looking for another human host to penetrate. Don't forget, it is the meracidia that penetrates the snail intermediate host, then it is the saccharia that penetrates the human host. Now, the meracidia has about 32 hours within the aquatic environment, after which, if it does not see a snail intermediate host to penetrate, then it dies off. But the saccharia stays for about 48 hours. That should be 2 days within the aquatic environment. After which, if it also does not see a human host to penetrate, then it dies off. And that means that the cycle will be cut off. But if it does see, then the cycle continues. This is the life cycle of Schistosoma. Now, it's also important we know that we have three different main species. Main species, and when it comes to diagnosis, the eggs of these three species have been carefully studied. The eggs of these main species have been carefully studied. We have the Japonicum, Japonicum. We have Mansoni. We have the Hematobium, Hematobium. So when we talk about the three main species whose eggs have been carefully studied, that is these three species. These three species. So during diagnosis, it is very, very easy for you to do the shape of their eggs to know the particular species of schistosoma that is implicated in such infection. Now, when we talk about pathology of this parasite, what does this parasite cause? The first thing we look at is what we call swimmer's itch. Now, at the point of penetration of this parasite, it causes a kind of rash, and that kind of rash is what we call the swimmer's itch. Swimmer's itch. Swimmer's itch. Itch. It causes a kind of rash or rashes that we call swimmer's itch. Apart from this again, the calcified eggs of this parasite that usually block the veins of the bladder may kind of add so much pressure on that vein such a way that it bursts, and when it breaks, now the person will begin to see traces of blood in the urine. And then when you see traces of blood, in fact, this is the most common sign of this infection. Once you see the blood in urine, that means the person now has a medical condition we call hematuria. Hematuria. Hematuria means blood in the urine. Then the person may also experience what we call portal hypertension. You know, the parasite, remember I said it also follows the the portal vein into the liver. Now, those parasites may add pressure on the portal vein. So the person will also experience portal hypertension. Portal hypertension. Now, apart from portal hypertension, it is also important we note that the person may also experience a kind of breaking of the varicose vein within the esophagus or the stomach. Yes, the vein within the stomach, esophagus or vein or the stomach will have a large vein that will call the varicose vein. Varicose. Varicose vein. Varicose vein. Varicose vein means a large vein. Now, if this parasite bloods and also breaks this vein, then of course the person may begin to vomit blood. Vomit blood. And this medical condition of vomiting blood is what we call hematemesis. Hema, hematemesis. Hematemesis. The medical condition where somebody begins to vomit blood. Okay, the person also experience a kind of inflammation of the liver. Remember, this parasite gets to the liver, and once it gets to the liver, if especially prolonged infection can result to a kind of inflammation of the liver, which is called hepatitis, with fibrosis. This is a very bad condition of the liver. So these are some of the things we're going to look at in pathology. Then what of diagnosis? How can this parasite be diagnosed? How can this parasite be diagnosed? This parasite can be diagnosed through the use of a demonstration of the presence of the eggs in fecal sample or urine sample. Don't forget, we said hematobium. Hematobium, sorry, is a species that is usually whose eggs are usually found in the urine sample, but the other four species I mentioned, the eggs cannot be found in the feces. So you get the urine sample, get the sample from the feces, and you mount it on a slide. Then using your microscope, you should be able to see the eggs of this parasite. That is what we call demonstration of the presence of the eggs in a fecal sample or urine sample, depending on the parasite you want to diagnose. Then, because we also have what they call the mucosal biopsy. When you now take a little tissue from the mucosal lining of the bladder, of the rectum, depending on the parasite you want to diagnose. That is what we call the mucosal biopsy. Mucosal, mucosal, mucosal biopsy. I don't know, this marker is not writing well. Biopsy. So you can see this is what we call the mucosal biopsy. So that is how this parasite this disease can be diagnosed. Then what again? We've talked about pathology, diagnosis, prevention and control. Government should provide safe water. mm, And apart from that, the river that is known to be contaminated with this parasite, government should try to build bridges so that people will not make contact with the river. Don't forget that it is only when you make contact with the river that the parasite will be able to penetrate through the unbroken skin. And people should not be defecating in water. If people don't defecate in water, it will be difficult for this parasite to the life cycle of this parasite to be perpetuated. People should not be urinating and defecating in water. These are some of the ways we can prevent and control this. Then epidemiology, what are the factors that help in the spread of the disease? Of course, availability of a water body that houses the snail intermediate host. That is number one. Then number two, availability of a snail intermediate host which is appropriate there, which is the appropriate host for this parasite. Of course, we have different species of intermediate hosts. We have one called the Onco melania, the Onco melania, Onco melania. We have Biome Falaria. We now have the Bulinus globosus. Bulinus globosus. These are some of the snail intermediate hosts that can carry this parasite. And like I said, we have three main species of this parasite. Each of these parasites has this as their snail intermediate host. Okay, treatment, and that's the last thing we'll talk about. How do we treat this disease, schistosomiasis? One of the ways you can treat it is through the use of drug. The drug of interest is mectizan. Mectizan. The some can also use what they call the praziquantel. Once you discover that an individual is suffering from schistosomiasis, then of course the person should be given this drug. This is where I will stop this class for today. Help me share this video to all the medical students around the world that are offering this course pathology. I know it will be useful to them. I remember I want to serve Dr. Bujon Uchenna. See you in my next class. Thank you.